A warm welcome to all the subscribers of Biotecnika. Today I have come up with a very interesting video which is developmental biology for CSI and life science from embryo to organism. We'll try to cover most of the basics involved in this process and also look at the points which are very important from exam point of view. Let's begin. Let me remind you to all the viewers that this topic is common with GATE Excel, which is life science syllabus as well. So don't skip this video, see all the basics and you can score some of the questions which come in the next GATE exam. First, let's begin with the basic question, what is development? Well, we have seen how a softie is built, isn't it? First, we have the ice cream. On top of that, we have the next coating. Then we have the sprinklers and then we have any wafers on it. So by this diagram, we understand that this development is also similar to this process, which is a step-by-step process. It is a slow process of progressive change. So remember that development is not only increasing in size. Well, it has to change something and that too slowly, one by one. That is simplest beginnings until final. What does it start with? It begins with a single cell, which is the fertilized cell or the zygote. And the zygote then divides mitotically to produce all the body cells. Well, the zygote, most of you would be already aware of, it is formed by the fusion of the sperm and the egg. And the zygote thus formed will be diploid in nature, whereas the gametes from which it is formed, it is haploid in nature. The zygote after that needs to produce many cells in order to form a multicellular organism. That is why the zygote divides mitotically just to increase the cell number to produce all the body cells. Most organisms never stop dividing. For example, if you take in our body, every day on a daily basis, we have the replacement of skin cells, we have the blood cell getting developed, and also in animals, there are processes in which development is involved, like metamorphosis or regeneration of severed or cut off parts. If you want to look at the entire embryogenesis process, what are the stages of development between fertilization and hatching, then we'll have to see step-by-step details. First process is gametogenesis, which is formation of gametes and precursor cells, which are also known as germ cells. Then all other cells which are present in the body, they are called as somatic cells, but these germ cells, they are gametes, they will be haploid in nature, whereas the other somatic cells, they will be diploid in nature. And germ cells only, they will be differentiating into the gametes, and the process by which they will do so is meiosis, which is the reductional division. As the germ cells will be diploid and we want to get the gametes which are haploid, so we have to reduce it to the N form, and that's why we need the process of meiosis, which is reduction division. The process of sperm formation is called as spermatogenesis, whereas the process of egg formation is called as oogenesis. I will show you some details of gametogenesis as well. Next, the process is fertilization, where the sperm and the egg, or the male and the female gamete, will fuse together to form a diploid zygote. So it is the fusion of the mature sex cells or gametes, and the fusion of gamete nuclei gives embryo its genome. This is from where the information will be present in the embryo to get the certain characteristics of structures which will resemble from the parental side also and from the maternal side also. Next, that we have our zygote. As I said, the zygote has to divide mitotically to get many cells for a multicellular organism, but this cell division, mitosis cell division in zygote, is a little bit different from that of the regular cell division. In a manner, simple way, if I say, they are very fast. So these special type of cell divisions, they are called as cleavage divisions. It's a series of extremely rapid mitotic division. That's one difference. Other differences I'll come to little later. And the zygote is divided into numerous smaller cells, which are called as blastomeres. After the cleavage division, the embryo will be forming a structure which is called as blastula. And the cells of the blastula is what is called as blastomeres. And that's why the zygote is divided into these numerous cells. Ultimately, it leads to the formation of the blastula. After blastula formation, the cells need to change their positions in order to form certain layers of cells, from which specifying the cells or giving cells their fate, what ultimately they will form. Because now you have the cells which all look same, which all behave the same. They don't have any specific gene expression inside them. But progressively, some cells will become neural cells, some will become muscle, some will become skin. So their fate and specification for it to be done properly, some are done early in the embryonic stage, even before gastrulation, but most of them will have to move and migrate to certain specific locations in order to achieve their fate by interaction with other cells. So gastrulation is the process where blastomeres undergo movements and they will change the positions. There will be extensive cell rearrangements. What is important from exam point of view is the type of movement that cells, individual cells or a layer of cells will undergo in order to achieve this process. And ultimately, after gastrulation, it will lead to the formation of something called as a gastrula, which is having 3 germ layers. Last part is organogenesis or morphogenesis, where cells interact with one another and they will rearrange themselves to produce tissues and organs, which ultimately will give rise to the adult form. So if you look at the pictorial representation, this is the zygote. It's just like a regular cell. Once it undergoes cleavage division, slowly it will form, one cell will divide into 2 cells, then the 2 cells will divide into 4 cells, 4 cells will be dividing into 8 cells. So since this entire embryo has 8 cells in it, that's why it is called as 8 cell stage. As I said, the cell divisions in embryo is a little bit different. One more difference is that most of the organisms will undergo cleavage divisions which are synchronous. Synchronous means they will happen together. If you see in your body, not all cells divide at the same time, right? But in zygote, in the embryonic cleavage division, most of the cells, early cells, they will divide together. That means when the zygote is dividing once to give 2 cells, both these cells now will divide together. So with one cell division only, you will be getting directly from 2 to 4. So it is an exponential increase. Okay. Now this smaller number of cells, if you have, let's say, 4 cells or 8 cells or 16 cells maximum, then these cells are very close to each other. They remain attached to each other and they resemble the fruit which is called as mulberry. You must have seen the fruit mulberry. It is like bunches of individual fruits are present together and it looks like a complete fruit. So that's why the mulberry word is morula in scientific terms. Okay, so morula, this 8 cell stage, in some organisms it is 16, some it is 4 cell stage, it is referred to as the morula. After the cleavage divisions, we form the blastula. The difference between morula and blastula, very important difference. So, my dear students, when you are preparing for CSI and NET, you should always focus, or any other PhD competitive exam, you should focus on keyword, and specifically in CSI and NET, because they have a tendency to play with words a lot. Okay, so morula, just knowing the physical difference is not enough. You have to know certain keywords. Morula is a solid ball of cells, which means that inside there is no cavity. But when we come to blastula, well, inside you have something called as a blastocele, which is a fluid filled space. So that's what is the difference between morula and blastula. In blastula, you have a cavity inside. Now, why this space is given, what is the significance of this space, you would understand in the next process, that is gastrulation. Well, if the cells are tightly packed into a closed space, then there will be no space for the movement of the cells. As I said, in gastrulation, the cells should move around, right? So that is why, in order to give certain space, the cavity is very helpful, and it also prevents the cells to interact too soon with each other. So as you can see in this gastrulation process, a layer of cells starts to go inside, and there will be other changes also. It is not so simple as it looks. and after that you will get something called as a gastrula with 3 distinct cell layers. And the cell layers, as you can see, this would be the blastocoel, which is now pushed in a very thin layer, and later in the development it also disappears. And this part which had slowly started to go inside, and it will create external external way also, in which there will be 2 openings for this particular tube. It is the endoderm, or we can also call it as the archenteron. Okay, so archenteron is nothing but it is the primitive digestive system. Later it will be forming the digestive system. So endoderm cells are the innermost cells. Endo word comes from the word inner. Okay, ectoderm is the outer layer of cells. Ecto word comes from the word outer. And in the blastocoel, sometimes, as you can see, these cells are attached in layer of cells, isn't it? In layers. Ectoderm and endoderm also. But there would be some cells which can leave the layer and they can come out in the space as loose cells. Okay, so those cells, they are called as mesenchymal cells. The cells which are present in the layer, they are called as epithelial cells, and the cells which type of cells which come out in the space, they are called as mesenchymal cells. These mesenchymal cells, they will form mesoderm cells, and from mesoderm you get all the connective tissue cells. For example, blood, then muscles, then skeleton, and other type of connective tissues. Okay. From the endoderm, as I said, you get the digestive system, and from the ectoderm you get the nervous system and the epithelial layer of cells. So as you can see, the cells have actually taken their position, from which fate specification will become very easy. Since this is the space from where the cells start to get inside, it created a pore in the blastula, or an opening in the blastula. That is why this part is called as blastopore. Okay, so this is the diagram of a cross section of a gastrula with distinct 3 layer of germ cells. Okay, so the organisms in which you have these 3 layers in the embryonic stage, they are called as triploblastic animals, where you also do have some diploblastic animals in which you have only 2 germ layers. Let me see who can answer me in the comment section some examples of diploblastic animals. Coming to the embryonic development, so you have the zygote, as I said, which divides into 2 cells. Then you will have the 4 cells, then 8 cells. Ultimately, you will have many cells, that is a solid ball. Then you have the blastula, which is the hollow ball, and ultimately you have the blastocoel, which is the cavity-filled or embryo. If you look at the different processes which take place with the zygote, well, we have growth. That means the ball of cells will increase in size till the cleavage division. If you will notice that the zygote and the blastula is more or less same size, it doesn't increase in size. So that is another feature of cleavage division. As I said, you will have to know the features and characteristics of cleavage divisions, as it is not regular cell division. So this is one more characteristic of cleavage division, that the cells don't grow in size. Okay, there's a reason why the cells don't grow in size. I will be telling you the secret very shortly. Then of course there will be cell division, where the cells will increase in size. Then we have differentiation, where the cell types will get diversified and they will form adult and the gametes later. Pattern formation, or which is like laying out the structure of the body. For example, the head part will be present somewhere, the thorax part will be present next to it, then the abdomen part. We can't have any cells present anywhere. So the cell types will arrange themselves to have a pattern in the body, and ultimately that will lead to morphogenesis, which is the generation of the shapes and the structures. Okay. Now first, talking about gametogenesis, as I said, meiosis process takes place over here. The process is divided into 2 sections. First, the germ cells are limited in number, so they will undergo proliferation just to increase their number, because the gametes will be produced in lot of number. And after that, there will be the first meiotic division, second meiotic division. So each mother cell, which is called as spermatogonia in males and oogonium in females, each mother cell will be giving rise to 4 gametes. Okay, but the difference in male and female gametes is that in spermatogenesis, one spermatogonium will give rise to 4 equal sized sperms, and all of them will be functional provided there are no mutations or any defect in the process. But in females, it is actually only one egg that we get out of one oogonium. Okay, the reason is because when the sperm and the egg will fuse together through the process of fertilization in order to produce a zygote, this particular zygote will have to support its development for few rounds until new genes and proteins are synthesized. So for some period of time, it is the egg cell only which has to take care of the entire embryonic development, okay, for the first few stages. But the sperm hardly contributes anything to the egg except its genomic material. There is no cytoplasm, sometimes mitochondria and the centriole they will be entering, but later the flagella also, but later they will be disintegrating. The centriole will only help in the aligning of the male and the female pronucleus, but after that even that doesn't have any role. So that is why the egg only has to support the development for the few early stages. That is why egg preserves its most of the cytoplasm rather than giving it away in the process of cytokinesis in the process of meiosis. Okay, so that is why in oogenesis you get one polar body and one secondary oocyte after the first meiotic division. Before the meiosis starts, it's called as primary oocyte in female and primary spermatocyte in males. Once this has divided, it forms a secondary oocyte and a polar body. The secondary oocyte will then again divide to finally give rise to the egg, and it will make another polar bodies. Okay, so one more polar body will be there. So totally you will get 3 polar bodies and one big egg with a big cytoplasm. So this is what is gametogenesis. Moving on to the events in fertilization. Well, fertilization consists of 5 very important events. The first one is that the sperm and the egg should move towards each other. So this process is mostly done by chemicals which are released from the egg, and the sperm will sense the chemicals and it will move in its direction. So this process is called as chemoattraction. It is achieved by some of the chemoattractants which are present in both invertebrates and vertebrates. For example, in sea urchin, a well-known chemoattractant is Rizact and Spiract. Okay, so they have their receptors which are present on the these are released by the egg. So obviously these chemoattractants will be received by the sperm. Okay, so these sperm, after receiving these chemoattractants, they will undergo some cell signaling. Generally the signaling receptors, they will be GPCRs. So after receiving the cell signal, after getting the cell signaling, the motility of the sperms increase and they will travel more faster towards the egg. Okay, so these are the soluble molecules secreted by egg. Then we have the exocytosis of acrosomal vesicle to release its chemical. We all know that sperm consists of the acrosome. It's basically the sperm head in which you have the nucleus, and on top of the nucleus there is a hydrolytic enzyme filled body which is a derivative of Golgi complex, and it is called as the acrosome. It contains hydrolytic enzyme so that when it meets the egg, it can dissolve the membranes of the egg. It can penetrate the egg membrane. Actually, egg doesn't have only plasma membrane, it has many layers, and so the acrosome has to dissolve the extracellular membranes of the egg. Then only it can fuse with the plasma membrane of the egg and it can go inside. So first of all, this exocytosis of acrosomal vesicle should take place so that it will release the contents outside. Then we have the binding of the sperm to the extracellular envelope of the egg. Okay, passing of the sperm through this extracellular envelope, and finally fusion of the egg and cell plasma membranes. So we have some fusogenic agents also, after which the nucleus will be mating and fusing together. Let me show you the interaction of sperm and egg in vertebrates. We also have in invertebrates, but we can't cover so much in one video. So here I am showing you only interaction of sperms and egg in vertebrates, where we have a very important layer of egg called as zona pellucida. So what is zona pellucida composed of? It is composed of 3 glycoproteins, namely ZP1, ZP2 and ZP3. And this is the egg membrane, whereas this is the zona pellucida. So as you can see, the sperm is first binding with the zona pellucida, addition to zona pellucida, and the acrosome is still intact. But now the acrosome is releasing its contents. First of all, it will bind with the ZP3 component and it will induce the acrosome reaction. After that, it will bind to something which is called as ZP2. Okay, so ZP2, once it binds to this component, the enzymes from the acrosomal vesicle will digest a slit in the zona pellucida from which the sperm swims to reach the egg surface. So once it is in the perivitelline space, through the equatorial zone, it is fusing with the egg membrane, because the fusogenic proteins are present in this part of the sperm. Okay, it is not present towards the head. So again, these are very minute things. The attention to details is very important in CSIR exam. Okay, so the fusogenic proteins are present in the equatorial margin of the vertebrate sperm, that is in mice model if you take. The membrane that covers the posterior part of sperm head, known as the equatorial segment, then fuses with the egg plasma membrane. Alright, so once the fertilization is done, there is a process to block the fertilization with other sperms. We don't want something called as polyspermy, right? Because it will disturb the ploidy level. That is why the eggs will have a mechanism to block the polyspermy. So one of the block mechanism that the egg has, there is some cortical granules present under the membrane. Actually, the cytoplasmic area which is present just below the egg membrane, it is called as cortex. And there are some hydrolytic enzyme filled sacs which are called as cortical granules just under the cortex. So the moment the fertilization takes place, this cortical granules will release their contents outside. Okay, so this is what is called as the cortical reaction. It also takes place in invertebrates as well. And one of the components of this cortical reaction is ovastatin in case of the mammals. Okay. So ovastatin is important, which clips off the ZP2. As I just said, ZP2 is very important for the fertilization process. If it is clipped off, then any other sperms will not be able to fertilize this egg. So based on all these topics, questions are framed, and after that, cytoskeletal apparatus of the egg will draw the sperm into the cytoplasm. Okay. Now, if you look at the interaction of sperm membrane with the egg membrane, so as you can see, this is the egg. This entire thing is the egg, right? A portion of it is shown where you have the egg plasma membrane, and you also have the zona pellucida on top of it. Now, outside the zona pellucida, there are some cells called as cumulus cells. These cells are released during the ovulation along with the egg only, just to protect the cell, egg cell. And sperms will travel through the through these cumulus cells and reach the zona pellucida, undergo acrosomal reaction, digest the zona pellucida, enter the perivitelline space and fuse with the plasma membrane. If you see the zoomed out picture over here, this is the sperm plasma membrane, this is the egg plasma membrane. You have several proteins or receptors components on the plasma membrane. For example, on sperm, you have the ZUMO, fertilin, iZUMO, fertilin and also cytidestin. Whereas on the egg plasma membrane, you have GPI anchored protein, CD9 and integrin. Well, these are fusogenic proteins. Fertilin and cytidestin are involved in sperm egg binding, whereas iZUMO and CD9 is essential for membrane fusion. The counterpart of iZUMO, which is present in egg plasma membrane, which it binds with it, is ZUNO. Okay, so JUNO and iZUMO, they have a lot of binding affinity with each other. In fact, we will be using this, exploiting this affinity in certain techniques in Unit 13 also, like affinity chromatography. Just how we have this biotin, avidin, some affinity, like that JUNO and iZUMO also forms a binding couple. So see how we are covering different aspects of one topic with respect to other units also. So that's how you should prepare for CSIR. You should be able to correlate with other topics in the syllabus as well. Okay. So these are the different egg responses after fertilization. Once you have the sperm binding or fusion to egg cell membrane, after that there is a tyrosine kinase receptor which gets activated in the egg, which will be activating the phospholipase C. If you have little bit of understanding of cell signaling, the tyrosine kinase can activate phospholipase C, which is a lipid digesting enzyme. It will cleave something called as PIP2, it is a phospholipid present in membrane, into two parts vis-a-vis IP3 and diacylglycerol. Diacylglycerol will be activating further protein kinase C, which will be activating sodium proton exchange, and as a result the protons will move out of the cytosol. Okay, so as a result there will be an increase in the intracellular pH, because if the H+ ions are moving out, inside the cell H+ ions will be less, and H+ ions is less means the pH will be increasing, as H+ ions are inversely proportional to the pH, right? IP3 production, on the other hand, will go and bind to the endoplasmic reticulum, where there are some ligand gated channels for calcium. So IP3 binds to that gated channels and it will release the calcium into the cytosol. Generally, the concentration of cytosol is kept low in the cytoplasm, because since it is a secondary messenger, too much concentration of calcium inside the cytosol, it can be toxic. But when calcium is released over here, it can signal the cortical granules present in the cortex of the cytoplasm, as I said, to undergo exocytosis. And one of those components, as I mentioned, ovastatin, like that many other components are there in the cortical granules, which will ultimately lead to the slow block to polyspermy and also the formation of the hyaline layer, so that other sperms may not be able to come and bind. They will remove the receptors, and plus it should be a thicker coating so that it can support the developing zygote further, right? The calcium release also needs to activate the cell egg cell so that it can start its cell division, and cytokinesis also should take place. That is why they will also activate for membrane biosynthesis. The mitotic cell cycle will be restored, and also it will stimulate the protein synthesis, DNA replication, etc. Okay. After that, there is the cleavage division. Once the fertilization happens, it is the first phase of embryonic development. You can see a micrograph over here in which we can clearly see that the cytokinesis is occurring over here. Ultimately, it will give rise to two daughter cells. Okay. I already mentioned the properties of cleavage division, that they are synchronous cell divisions to decrease the cytoplasmic to nuclear volume ratio. The size of the zygote, the embryo, it's not changing. It's just that right now you have a very big nucleus compared to the cytoplasm. Okay, so that is why we need to divide this ratio so that you have some good balance of the cytoplasm and the nucleus. Okay, so cytoplasmic volume does not increase in initial phase of development, it just gets divided. So the same cytoplasm only gets divided over here. The point is that this one nucleus cannot support the cytoplasm, such a huge amount of cytoplasm. Okay, so that is why if it can divide into individual cells, so the cytoplasmic content will be less. Very little gene expression during cleavage divisions will be taking place, and that is the reason why the cell does not increase in size. And the secret which I told you, the reason why the cells does not increase in size is because the gap 1 and gap 2 phases which we find in cell cycle, they are not present in cleavage dividing cells. Okay, so that is why we have very rapid cell division. Why it is very rapid? The entire cell cycle, roughly it will have some 16 hours or something in adult cell, but here it will hardly take some 30 minutes or so. Okay, so the reason is because in somatic cycle you can see the G phase, G2 phase, okay, it is occupying a lot of time, even the S phase also. But in cleavage division, you only have the S phase and the mitotic phase. Cell fate determination and access specification may happen during this stage only. Okay, so this is an example of the early cleavage in xenopus or amphibians. where you have the first cleavage division going meridionally. Meridionally means along the axis of animal and vegetal pole. And second cleavage is also meridional. Third cleavage, as you can say, it is equatorial. So like this, many planes of division will be there, ultimately producing a morula and a blastula. So there is this blastocele also. Two functions of the blastocele: it will prevent the cells from interacting too soon, and it will allow space for cell migrations during gastrulation. So here is actually where the gastrulation process starts, right? Now there is some different classification of egg based on the amount of yolk. Like different types of animals have different amounts of yolk in their cells, and that will affect the cleavage division. For example, isolecithal egg. The word lecithal refers to the amount of yolk. So when you have iso, means it is uniformly distributed inside the cytoplasm. Okay, so there is very little yolk actually. Ignore the dark color of the egg. It is just to show that it is uniformly divided. Then you have alecithal, where there is very little or almost no yolk present. So this is what we have in the mammals. This type of egg we have in the sea urchin. You should also know the representative organisms in which this type of yolk will be present. Then we have mesolecithal, which is the moderate amount of yolk and concentrated at the vegetal pole only. And then we have the heavy yolk content eggs, which is in insects and which is in birds. Okay, so mesolecithal is found in xenopus, whereas this insect and bird, they will have macrolecithal egg, means there is a lot of amount of yolk. In case of insect, this yolk is concentrated only in the center. That's why it's called as centrolecithal. In case of birds, this yolk is present towards the vegetal side, and that's why it is called as telolecithal. So bird, fishes, reptiles, they have huge content of yolk, hence they are called as megalecithal. So this is a chart showing the same, and the representative organisms, you must be knowing all this properly. Okay, and as I said, based on the type of yolk, you have the cleavage division. For example, if you have a alecithal egg, isolecithal egg and mesolecithal egg, as I said, they have little amount of yolk. Hence, the cleavage division is able to cut the entire embryo. Okay, so entire embryo means the word we are looking for is whole, and that is what is called as holoblastic cleavage. Holoblastic cleavage is the complete division of egg, and the cleavage furrow extends through the entire egg. In holoblastic cleavage, the furrow squeezes around the periphery. So you can see the cell division is happening from the periphery over here, just like a belt tightening, and the two cells will be pinching into two cells, right? The cells will be pinching into two cells. Another one you can imagine where there is huge content of yolk. So yolk is a hindrance, or it is an inhibition to the cleavage furrow. It cannot cut through the yolk. So wherever you have huge amount of yolk, the cleavage furrow will not be able to proceed forward. So it will only cut like a knife, not from both ends. Okay, so in meroblastic, this is called as meroblastic cleavage, or which is also called as incomplete cleavage, right? So incomplete division of egg, only a portion of cytoplasm is cleaved. Furrow starts at the animal pole, progresses into an egg like a knife, and it will stop when it encounters the egg. So this takes place in telolecithal and centrolecithal egg. So as you can see, these are the different types of cleavage divisions that we have in organisms. For example, in sea urchin, it has isolecithal egg, hence it undergoes holoblastic cleavage, where the entire embryo has been divided. Mesolecithal egg, also it is holoblastic. There is some amount of yolk at the vegetal side, but still the cleavage division is able to cut through, although the vegetal cells are little bit bigger in size than the animal pole, because animal pole doesn't have yolk. Whereas telolecithal egg, you will see the cleavage division is only at the top portion of the egg, which is devoid of yolk. And whereas in centrolecithal, since the central part is completely filled with yolk, so the cell division cannot happen at all. So in insects, the cell division will not happen until the 14th stage. So until 14th stage in insect, it is just present in the form of a syncytium. Okay, so in syncytium there is only nuclear division, but not cytoplasmic division, right? It will start only at the 14th cell division. So I hope you got an idea of how the zygote slowly progresses to the adult organism. As I said, from here you will have the gastrulation process, from there you have morphogenesis, organogenesis process, ultimately giving rise to the adult. Follow Biotecnika classes and videos for more information because we have a lot of details to cover under this topic. See you in the classes. Thank you everyone.